Okay, let's start today's session. So this is week 7 or 7th assignment. So in this week you have studied mainly two things: citric acid fermentation and lactic acid fermentation. And in those lectures you have studied about what is the requirement, what is the microorganism used, what are the different conditions, what are the applications of these two different type of fermentation. So today's assignment is based on those two fermentation. If there is any issue regarding my voice or screen, please let me know. So coming to the first question. Okay, let's read it. Which of the following bacterial genera is unsuitable? Okay. Which of the following bacterial genera is unsuitable for lactic acid fermentation? Uh, Now it's fine, Satvika. Okay, let's start. So what are the options they have given? That Pediococcus, Lactococcus, Streptococcus, and Staphylococcus. So they have mentioned all the bacteria from this cocci. This is the shape that is representing here. So it means all these bacteria have same type of shape. Mainly coccus represent the circular kind of shape. But they are asking which is not suitable for lactic acid fermentation. Okay, Satvika, yeah. Yes, this is correct. Staphylococcus is not used in lactic acid fermentation. Let's read a little bit more about this lactic acid fermentation or lactic acid bacteria, especially LAB, that we represent in a short form as lab. Because in the previous session we have discussed that there are four major components of any fermentation process. So one of that is the microorganism that is used. So in this is lactic acid bacteria. So what is lactic acid bacteria? This is mainly belong to this lactobacilli, and you must know the properties of the bacteria or the microorganism that is used in that fermentation process. So you must know few things regarding that microorganism, that what is the type and what is its family, whether it is anaerobic or aerobic, what is the especially the conditions that is required for its growth. This is really important for this fermentation process, because only that you will be able to prepare or ferment anything properly. So the properties of this is that it is a gram positive bacteria and non-sporulating. Non-sporulating means it will not form spores after like its growth. It won't form a pores like structure, because some microorganisms are like when they grow on a pet. Suppose if you grow on a lab and they have grown on a petri plate, then when you observe that plate you can see the pores like shape, not shape but they form pores. So that is we called as sporulating, and non-sporulating they won't form that type of structure. And anaerobic means that it does not require oxygen for its growth, because some microorganisms are like that you should provide them oxygen, otherwise they won't grow. But for this lactic acid bacteria it is not required, and even if you provide the oxygen it will grow. But so it is a oxygen tolerant also. And these are the different microorganisms that are belong to this lactic acid bacteria or lactobacilli family. That is lactobacillus, leuconostoc, pediococcus, lactococcus, streptococcus. Okay, so this is mainly regarding this lactic acid bacteria. And the pH, this is really important for every microorganism. As if you change the pH anything like from 7 or 8, then this won't grow. Similarly happen if you go below 3. Usually microorganisms don't grow below 3. Only very few are able to survive at that very acidic pH. So few things are really important for any microorganism. Okay, for this question answer is D, because this staphylococcus does not belong to this lactic acid bacteria family. Okay, these are, okay, I will clear this. These are some of the like applications of this lactic acid bacteria, that it forms chemicals, that is lactic acid, also used as a starter culture in food industry, pharmaceutical delivery system, in making especially the drug delivery system or vaccine, this is used. So it has a major applications in different fields. Okay, so as I have mentioned that it is a gram positive, so you must know a little bit of this, that what is gram positive or gram negative. There are two type of, we mainly divide bacteria in a two categories, that is gram positive and gram negative. So those who are from biotech background, they have studied this, but I will tell you a little bit of this, that this is a gram positive bacteria, this figure, and this is of gram negative. What happens, suppose this is your bacteria and this is outer layer or outer covering of this, that is we called outer membrane. So the structure regarding this outer surface is different in both the type of bacterias, gram positive and gram negative. So and when we stain any bacteria with gram staining, or you consider it as a dye, like we want to know the bacteria is gram positive or gram negative, so we use this gram staining technique. It is a kind of dye, we use two different dye in that. So what happen when we use this and it will give violet color, then it will be gram positive, and if want it won't give violet color, either it will give pink color. So this is gram negative type of bacteria. Oh, why they are giving different color with this same staining? Because of this structure, because when initially the dye was enter, because of this like major presence of this peptidoglycan layer, that is yellow in color, it will block the color and it will give finally violet color. But when the gram negative bacteria, it is very small layer or a single layer of this peptidoglycan is present, due to which it is not able to bind this violet color, and this will further stain with another color, because of which it get pink color. Okay, so understood this, at least you must have basic idea of this now, that what is the gram positive bacteria and gram negative. Although it is not exactly in your course, but when you study about the microorganism, then you must know few properties regarding them, like what is the gram status of that bacteria, and whether it is anaerobic or aerobic, pH, and the other family members. Okay, let's move to next question. Which of the following component is are essential for the growth of lactic acid bacteria, that we are discussing? That now they are asking about the growth factors, or you can, what are the different components required for its growth. So they have given options: vitamin C, vitamin B, amino acid, or peptic acid. So anyone wants to answer? Okay, vitamin B, yeah, correct. Any other? Anyone wants to say like what is required for this lactic acid bacteria? Okay, vitamin B is correct. Yeah, vitamin B is required for its growth, but amino acids are also required. So for this lactic acid bacteria growth, both the vitamin B and amino acids, both the components are required. So for this answer is B and C. Now let's move to next question. Match the following grade of lactic acid mentioned in column A with its corresponding strength mentioned in column B. So what they have given? They have given that different grade. Grade means that component have different percentage of that compound, that it is saying the lactic acid, so it means these three grades contain different percentage of lactic acids. So let's read. First one is crude or technical grade, second option they have given edible grade, and the third one is US pharmacopoeia grade, means pharmaceuticals. In pharmaceuticals, what percentage of this lactic acid is used, and in other like for edible, which is suitable. So anyone wants to answer this? And apart from these three, there is one more grade of this lactic acid. So lactic acid is present or found in four grade. So let's discuss. First one is crude or technical grade. So this is mainly used in textile industry. Okay. So how much percentage is required in this? This contain this 20 to 80% of lactic acid. Okay. In edible grade, in few food items it is present. Then this is clear that US pharmacopoeia means pharmaceutical industry contain this. So for edible grade, this is 50 to 80% of lactic acid is required, and this US pharmaceuticals in this 85% of lactic acid is required. And that one more grade, what it is called as lactic acid, is also used in plastic formation. Okay. So in plastic formation, it also contain around 50 to 80%. Okay. So you must know the application of these four also, and their percentage also, the strength of lactic acid. So for this answer is, for first it is A, for second it is B, in the pharmaceutical it is C. So the answer is 1A, 2B and 3C. So for this question answer is A. Yeah. Now next question is, if I'm too fast or too slow, please let me know. Okay, let's read the next question. Other than lactic acid, which other component may be produced during lactic acid fermentation? So anyone should answer this question, because it is a simple question and you must know what is the byproduct that is formed during this lactic acid fermentation. So tell me whether it is 1,4-butane diol or 2,3-butane diol, or both these components are produced. Okay, Satwika, yeah, correct. that this 2,3-butane-diol is formed as a byproduct during this lactic acid fermentation. So this is also important. You must know what is the byproduct that is forming in any type of fermentation and in fermentation of any product, because it is not possible to form a single product during fermentation. Byproducts will always form. So you must know about them. So during this lactic acid fermentation, 2,3-butane diol is formed. So answer is B. Okay, let's read this question. The advantage of using lime as a neutralizing agent in lactic acid fermentation is R. So what are the options they have given? First one is shorter fermentation time. Second option is higher yield of lactic acid. And the third is prevention of bacterial inhibition by acid formation. Or option D, all of the above. So all of you know that lime is used as a neutralizing agent in lactic acid fermentation. Okay, first of all, tell me what is the form or what is this lime?
calcium hydroxide.
Yeah, correct. This lime is calcium hydroxide that we used in lactic acid fermentation as a neutralizing agent. So what are the functions of this? That it shorten the fermentation time, correct, because it will precipitate early. Then higher yield of lactic acid, this is also correct. And the prevention of bacterial inhibition by acid formation, this is also a function of this lime. So for this question, answer is D. During lactic acid fermentation, you have added lime and other reagents also for other different type of function that we will discuss in the coming questions. Okay. Yeah, correct answer, Sarvika. Answer for this question is D. This perform all these functions. Yeah. Okay, let's read this. Whey is a major raw material for lactic acid fermentation. It is the byproduct of dairy industry. It contains the following. So what are the components that is present in whey? They have already told this, that this is the byproduct of dairy industry. Like when you remove the cheese and other part from the milk, then the remaining part is called as whey. And this whey contain, what are the major components of this whey? What are the options? Albumin. B option they have given. Sodium chloride. Third one is calcium lactate. D is lactose and vitamin G. So this whey contain this albumin protein, correct? So sodium chloride, no, this is not present. Calcium lactate, this is also not present. But this lactose and vitamin G is also present in this whey. Lactose and vitamin G. And this is also used as a raw material for lactic acid fermentation. So for this, answer is both A and D. Yeah. Let's read this question. Match the components mentioned in column A with their function mentioned in column B. So these are the components that we add during the lactic acid fermentation, and in the column B they have given the functions like what it is used for. So you have to match these. So let's read. First one is slake lime. As we have already discussed that what it is.
Neutralizer.
Yeah, correct. Neutralizer or a neutralizing agent. Then second option they have given. So for first, this is matched with C. Then second is they have given is riboflavin. Third option, third column is activated carbon, and fourth one is nicotinic acid. So we know that usually, like if you know the first one, then you can mark, but you must know the function of others too. This activated carbon, this you have studied or have seen in many advertisements also. What it is, mainly activated carbon is used for purification or removal of particles. So it means it is mainly used for removal of anything that we use in different processes. So for here, what it is, it remove the color, so it is called used as decolorant. Okay. Then riboflavin is a vitamin, and it is used as a precursor, or you can call it as a used in growth components or growth factors. So it is a activator, and this nicotinic acid is used as a stimulant during this process. So for this answer is A. The slake lime is used as neutralizer because it will neutralize the citric acid initially we precipitate it with. We will discuss this whole process in detail, but initially it is used this. And the activated carbon, for wherever it is used activated carbon, not only this fermentation, even different processes, it is mainly the main function of this activated carbon is removal. So it is used here as a decolorant. Then riboflavin we have discussed used as activator, this is a vitamin B, and nicotinic acid as a stimulant. So for this question answer is A. Oh. So in the previous session you have studied two type of fermentation, that is lactic acid fermentation and this citric acid. This lactic acid fermentation, which microorganism is used? Bacteria. Which bacteria? And in this fungi is used.
Aspergillus niger, ma'am.
Yeah, Aspergillus niger is used for the citric acid. Although other different fungi are also searched or researched, but this is the most or majorly effective or majorly used because the efficiency of this Aspergillus niger is more than all the other fungus that have found till now. So for this, let's read the question. In citric acid fermentation by Aspergillus niger, if pH is more than 6, then which component is formed instead of citric acid? So what are the options? First one is gluconic acid, second one is oxalic acid, third option is citric acid anhydrous, and the last option is none of the above. As I have mentioned earlier that pH plays a major role. All the growth factor or growth components are really important during any fermentation. Okay. So as it is seen here, that is they are asking for pH more than 6. So can anyone tell me what is the optimum pH for this citric acid fermentation? Like what is the fermentation required? What is the pH required for this citric acid fermentation? Because then only we can discuss like whether this pH 6 is more from the optimum pH or less than. No one remember this pH?
3.5.
Yeah, correct. This is 2.5. And this is very rare pH you will find at which microorganism grow. So at this pH we have advantage of this also, that this pH will not allow the growth of other microorganism. So for this chances of contamination is very less because of this pH value. So now they have asking what happen when the pH is more than 6. So what happened, like there are two type of changes occur when pH is more than 4. Okay, let me write in a different way. Okay, optimum pH is 2.5, and for this, if pH is more than 4 and less than 6, these are the two conditions during this fermentation, and there is one condition when pH is more than 6. So what happened when pH is between these two, this citric acid, instead of formation of this, what happened, gluconic acid start forming. But when pH is more than 6, like it will go to basic, then it form the oxalic acid. Okay, so you must know both the conditions. So for this, answer is B, because they are asking more than 6. Yeah. Okay, let's read next question. Iron acts as an important cofactor in the TCA cycle for which of the following enzyme? Okay. What are the options? Fumarate, aconitase, isocitrate dehydrogenase, and none of the above. So first we must know what is this TCA cycle. Okay, this is the details of this is not in the not the part of the course, but this is like the formation of this citric acid. This is through this tricarboxylic acid cycle, or it is also called as Krebs cycle. Those who have studied the course biochemistry or from biotech background, they must have studied this. So this is a like chemical. In this cycle you will study step by step formation of this citric acid. That's why the name is tricarboxylic. Because when you make the structure of the citric acid, you will have 3 COH group. So this iron normally, that, yeah, iron is a cofactor. What is this cofactor means? That is required for, suppose this is enzyme, any enzyme that is required for the reaction, so that will help in its activity or increase. So different components are required for that. That is called as cofactor, and this iron is a cofactor of aconitase. This TCA cycle I have also mentioned in the coming slides, but you don't need to learn it step by step, but at least have a little bit of idea of that, like what happened in every step, what are the different enzymes. So for this question, answer is B. Yeah, yeah. This is called as TCA cycle. So this part is glycolysis, that glucose, or when you take any sugar component that is converted to pyruvate, then the further it form acetyl coacetate, and it further form the citric acid fermentation or citric acid formation, not fermentation exactly. So it this is the enzyme that aconitase is required for the conversion of citrate to isocitrate. So different components are required at the different steps, and this those if want to learn more, you can focus more these things, what are the like other components that is required this NADH is forming here, and what are the steps CO2 is required, CO2 is forming or not forming this GTP. These few things are, you can have a look at once at the every step of this. And those who have studied or who will study the biochemistry, they must learn the every structure of these also, and the function of every enzyme, what are the different cofactors required for every enzyme. But for this course you don't need to study in detail this TCA cycle, but have a brief idea of this. This is representing that citrate has 6 carbon. This 5C, it means this structure has 5 carbon. Okay, and these are the enzymes mentioned here. So this is the next question. Which of the following enzymes accelerates the growth of a Aspergillus niger when grown on a solid surface? Yeah, correct. Answer is option A, this alpha amylase. So you have seen in this assignment that the mostly questions are related to the what are the growth factors required, what is the microorganism or how much it is forming. So all these things in the fermentation process is essential, or you can call it as like optimum. If everything is at optimum, then the percent of product formation will increase. So that's why we need to learn about every major and small changes in the fermentation process, because whenever you change even a simple factor, then the whole process will change and the productivity will decrease. So that's why you need to have everything at a proper space. So for this question, answer is A, alpha amylase is required for this solid surface. Okay, let's discuss a little bit of this, what is solid surface. And like they are discussing about the solid surface using this Aspergillus niger, means they are talking about the citric acid fermentation. Okay, so normally citric acid fermentation occurring at a two step or in a two different types, you can say. First one is surface related, second one is submerged. So on the surface, this is mainly a either you have the solid surface or liquid surface. In submerged you have different type of fermentation, that either it is stirred reactor or airlift, that they are talking about the large fermenters. So mainly what happened, around 80% of the industrial this citric acid fermentation is occurring using this submerged, but on a surface based that is around only 20%. There are some advantages of this also, and the same for this. But when you need to make it at a larger scale, you prefer the submerged fermentation process as compared to surface. Like in solid and liquid surface, what happen in solid, you will take the growth culture and the Aspergillus niger as a solid layer. Means you grow the Aspergillus niger suppose on a plate, then you will take a layer of that Aspergillus niger. But when you go for the liquid surface, what will you use? You won't use the whole layer of that Aspergillus niger that is growing there. You will take the pores. We have discussed earlier na pores. So this is the microorganism that produce majorly pores, and in a large quantity. In a very large quantity, it will form the pores, and this pores is directly mixed in the liquid solution. So these few little differences is there, and this you can come to know from the name itself, that it is a solid surface and this is a liquid surface. But this is used at very few places. But there is one major advantage using this, that when you use this surface fermentation or the surface method, what happen, it will not reuse or waste the metals. Like suppose some trace metals are present, so that are consumed in this process, but in this they are not properly used. So that's why in this method preferred this is preferred because of this, that different metals are already consumed in this method. So for this question, answer is A, that alpha amylase is required for this solid surface. Okay, next question is, let's read it. Citric acid is separated from the fermentation broth by using, first one is sodium hydroxide, second option is potassium hydroxide, and the third is calcium hydroxide, and D is none of the above.
Calcium hydroxide.
Yeah, correct. As we discussed that lime is used, this is lime that is used for the separation of this citric acid. So for this answer is C, but let's discuss this process. Yeah, that suppose you have here your, this is the bioreactor in which suppose citric acid is forming, and at the first step, like after providing every growth vector and the other things, that initially you use lime. So what will happen when lime is used, it will help in precipitating, and it will increase the concentration as compared if you won't use the lime. So I will mark the major things that is required or that you must know. This is the whole process or the downstream processing of this citric acid. Okay, this figure representing this. Then at the second step, after washing, what you use the sulfuric acid. Okay, what happened when you use the sulfuric acid, that gypsum formation occur, means if any waste gypsum is there, it will remove, and the filtered part we used for further purification part, like this ion exchange, it means this is a chromatography method they are using. Then different processes are used in the whole downstream processing, like here they use the ion exchange for the separation, then evaporation is also used, then crystallization, means the formation of crystal is occurring at this step. Then after that, separation. For the separation, different processes can be used. Can anyone tell me the name of the separation process? Any separation process?
You mean membrane or
Yeah, correct. Membrane is also used. Membrane filtration, centrifugation,
Ultrafiltration.
ultrafiltration. Yeah, correct. That is also used. So these different type of separation methods is used. Then further, after the final filtration, if you have the final component, then the drying will occur, and finally the packaging. Few major things you will find in every downstream processing. Few things will change regarding the component, but few things like separation, then which method you are using, that will depend upon your component, but separation method, and suppose this is also separation, and this crystallization, evaporation, drying, packaging, these are the major steps or common steps you can say in every downstream processing. Suppose this is we are discussing about citric acid. So what we are using? We are using here lime, sulfuric acid. But if it will go to the lactic acid, then these two components will change. And if you go on any other like alcohol, then it will change. So few things will change according to the component, but few methods will same remain same. Suppose here you have used ion exchange, but for lactic acid you might use any other type of chromatography, or you might not use chromatography. So this will little bit change related to the product you required. But this drying, packaging, all these things are common. So this is related to the citric acid fermentation. Okay, yeah. This is the major reaction that I'm trying to show you here. That when you use the lime, what is the reaction occurring, and when you use the sulfuric acid, how it will change the composition of this citric acid. Okay, this is the citric acid, or hydrated, in a hydrated form that is present. So when you use this calcium hydroxide or lime, this calcium citrate tetrahydrate form, like it will precipitate this. Then after that sulfuric acid is used. So when you use the sulfuric acid, you can see the difference here, that calcium and this remove this calcium and water component from the structure, and it will only contain dot one H2O, or only one part of this. So if you need it further, either you can use in this form only, like in a dot H2O, or in a hydrated form. You can also use it in a anhydrous form. So for both these two, you can use a different separation process. Okay, so this is the major reaction that is occurring. Everyone understood this process? Like you can't learn everything for the every fermentation, but you must have a basic idea like what are the processes that is present in downstream processing. Okay, now coming to next question. This calcium citrate reacts with sulfuric acid to produce citric acid, and what is forming here? Just we discussed in the previous slide.
Gypsum.
Yeah,
Gypsum.
gypsum, correct. That just gypsum, for this removal we use this, or this is formed as a side product, you can see. So for this, answer is C. This question we have also discussed in that slide. Let's read it. Before the centrifugation process, the major downstream processes in citric acid industry are: first option they have given decolorization, second is crystallization, and C both A and B, and D none of the above. So now anyone please answer.
Both A and B, ma'am.
Okay, anyone?
Only decolorization.
Okay, okay, let's go back to that slide. Yeah, this is here. So they are, what they are asking, they are asking for the centrifugation process. This centrifugation means that is separation process. For the separation they are using this centrifugation method. So before the separation, this decolorization, crystallization. So yeah, correct that crystallization is occur before that, and decolorization is also occur, because for decolorization we have used the activated carbon that we used initially in this process itself. So both the things, that decolorization and crystallization, both the process occur before the separation, because at this stage we have final product in the purified form, or whatever percentage that we required. Suppose you want to make 50% purified, So you have at this step, because after this only drying means you are forming, you are putting that in a vacuum for different application or packaging. So in these two steps, like after separation only these two, drying and packaging, occur. So all this process, crystallization and decolorization, occur before that only. Okay, so for this answer is C, both A and B. Both the processes occur before this final separation. Okay, now let's move to next question. Now I don't think so. I need to read this. Okay, citric acid is produced through which cycle?
TCA cycle.
Yeah, correct.
PCA, ma'am.
Okay. So for this answer is B. So this is the last question for today's assignment. Let's discuss this. What they are saying, citric acid is produced in a submerged fermenter of 150 meter cube capacity using Aspergillus niger. The average time of fermentation is 120 hours and citric acid yield, just a minute. Yeah, now coming back to the question. Citric acid is produced in a submerged fermenter of 150 meter cube capacity using Aspergillus niger. The average time of fermentation is 120 hours and the citric acid yield is 80% weight by weight. If the idle time of the reactor is 10 hours, what is the productivity of citric acid? Assume the initial sugar concentration is 100 gram per liter. This is also a very simple question. But first you must know what is this idle time and what does this mean that 80% weight by weight that yields. Okay, like if it means 50% weight by weight, or that yield is 50% weight by weight, it means if you have taken the 100 of substrate, only 50% or 50, suppose if you have taken 100 kg, from 100 kg only 50 kg of the product is forming. It means it is telling that how much yield is forming from the initial substrate. It's not like you have taken 100 kg of substrate and 100 kg of the product is forming. If this happen, then it is 100% weight by weight yield. Okay, now understood the meaning of yield. And what is this idle time meaning? You must have used this term idle at different places also. So what is this? Anyone want to discuss this? What is this idle meaning?
The time the process take to occur.
For the process that occur. Okay, but they have given that average time of fermentation is 120 hour. No, that this is not the process time. Actually, this is the time
It's
that
in
is.
the reactor.
Reactor is closed that time. Reactor not working on that time.
Yeah, correct. Reactor is closed at that time. That time is used. That time is called as idle time. Means suppose you are running a fermenter. So you can't run it 24/7. So suppose you have to run a fermenter for 30, suppose 38 hours. So your fermentation will run, suppose it will run only 12 hours. So after 12 hour running, it required 2 hours of break. Similarly, it will run 12, then it will take 2 hours of break. So this 12 hours, it will, when you count the total 38 hours, this time you will take, but this time is also going in this. So this is the time, that time reactor is not working, that time is called as idle time. Okay, understood the meaning of this idle time? Or you can say, like, suppose you are working in a any, suppose you are in a class, but you have take class timing is like for 50 minutes, but in that between 10-minute break, so that time is called as idle time. Like, that time productivity is zero, that time we usually called as idle time. Okay, so now you understood the question, that citric acid is produced in a submerged fermentation. So total volume they have given that 150 meter cube, that this is the size of fermenter. Then the average time of fermentation is 120 hour. That time reactor is working and the citric acid yield is 80%. Then if the ideal time is 10 hour, what is the productivity? And they have given the initial sugar concentration is 100 gram per liter. In simple terms, what is productivity means? This term we use like in our everyday life itself, that today we have done productive work or not, or we have worked only for 2 hours, productive. So it means how much that total, the productive time, or the, you can say, properly used time to the, as compared to the total time. Similarly, what happened in this productivity, how do we define it? That total citric acid formation, that is forming, and the total time taken. Okay, these two things are required to find the productivity. This total time is this sum of this, because for our, that this is taking the total time. Suppose this is 130 hour. Okay, so now we have to find this total citric acid total citric acid amount. From where we will find this? From these equation, or from this information. This 100 gram per liter, if you convert in kilogram per meter cube, because the initial unit we have in meter cube and this is gram per liter, so it won't work. We must need both the things in a similar unit. So this 100 gram per liter, anything which is gram per liter is equal to kilogram per meter cube. So everyone understood this common conversion, that when you change the gram to the kilogram, it will be 1000, and from liter to meter cube it is also 1000. So that's why if any unit is given in gram per liter, it is same as in kilogram per meter cube. So now we will find how much citric acid is forming. So let's, that initially we have 100 gram per liter, or now you will say 100 kilogram per meter cube. This is the initial sugar concentration. Okay, then how much is the yield? That from the 100, how much it is forming? It is forming only 80. So either you can write it as a 80 out of 100, or you can write it as a 0.8. Okay, this is from this initial sugar concentration, but total is 150 meter cube, that is the total volume of the fermenter. Okay, So this is the total citric acid concentration or amount. You can call it amount of citric acid is forming is this. So when you solve this, it will come around 12,000 kg. So everyone understood this, that how we are calculating the total citric acid?
Ma'am, can you repeat that again? The process.
Yeah, correct. Okay, I will repeat. We know that citric acid is forming from this, like you need a substrate, so that is 100 initially, and the yield is 80%. Means if 100 from 100 gram only 80 gram is forming. This is the meaning of this 80% weight by weight yield. Then we have the total submerged fermenter. Means this reaction is occurring in a large fermenters with the total volume of this. So we will, that from 100 it will form only 80%, and the total amount is 150 meter cube. So when you multiply these two, this total citric acid amount we have. Now
Okay, ma'am. So
understood.
this is from 100. This is for 100 grams, ma'am. Like for from 100 grams, this much amount of kg you're producing.
Yeah, initial consent, this is only the concentration, but the total amount, when you will say this is the total amount when you multiply it by 150 meter cube,
Okay.
because that is only, like, this is the amount we are talking about here.
Ma'am, like what is the purpose of conversion, ma'am? This like 100 into 80, this thing, this process.
This is not the conversion, this is the total amount of citric acid produced.
Okay, ma'am. Okay.
This is not the conversion. Conversion is when you convert this gram per liter to the kilogram per meter cube, it will remain same. Means 1 gram per liter is equal to 1 kilogram per meter cube. So for this 100 gram per liter will remain as 100 kilogram per meter cube. Okay. So from here we calculated the amount of Ca produced using this fermenter. So it will come around 12,000 kg. And the total time we have 130 hours. So what is the productivity? Productivity is ratio of these two, that total citric acid by the total time taken, that 12,000 kg by 130. Okay, so when you calculate this, how much it will come?
92.307.
Yeah, correct. This will come around 92.3 kg per hour. Okay, now understood this question, that the productivity of this is 92.3 kg per hour. If anyone has any doubt in this question or any other question. Okay, no doubt.
Ma'am, like I have one doubt, ma'am.
Yeah.
Ma'am, what is the purpose of riboflavin, ma'am? Like in this fermentation process, riboflavin or
Okay,
any vitamin?
riboflavin. Yeah, that is required as a different, you can say it as a growth factor, like for the growth of any component or any bacteria, any microorganism, you require different things. Like for our, suppose for our growth, it is required different metals, required different vitamins, different amino acid. So similarly for the microorganism, all the different components are required for their growth. So riboflavin is used similarly.
Okay, ma'am. So it is acting as an activator, ma'am? Activator kind of
Activator
thing, or
means like it will help in growth of that bacteria, and That is important for this lactic acid fermentation, no? Lactic acid bacteria. So it will enhance the process.
Yes, ma'am.
Anyone has any other doubt? Okay, I'll stop the recording.